Hi everybody, my name's Kathleen Dunbar and I'm a final year PhD student at the University of Surrey, and I'm going to be presenting an overview of my work on hydrogen production from E. coli and their uses within biocoatings. So why hydrogen? Well, in order to combat climate change and global warming, we need to look at some alternative, cleaner, more sustainable energy sources. And hydrogen is a great alternative because it's a very good energy carrier, and when combusted, it only produces water. However, 96% of hydrogen globally produced at the moment is actually made from a fossil fuel source. And the 4% that is classed as green, where we use electricity to split water into hydrogen and oxygen, is very expensive. And also, if your electricity is being made from fossil fuels, can you really class it as green? So this is where biohydrogen comes in. So biohydrogen is the production of hydrogen from a microorganism. There are two main methods, the photosynthetic method and dark fermentation, and I'll explain a little bit about each of them. So in the photosynthetic method, organisms such as algae and cyanobacteria will use sunlight to convert water and nitrogen into oxygen and hydrogen. There's a few benefits to this, obviously the one being that your energy source is the sun, and you don't need to put in that many additional nutrients. However, if you want this to be an efficient process and to run for 24 hours, you actually need to put in some artificial sun, because obviously sun doesn't shine for 24 hours. So you need to have that artificial light source. However, if you had one cell on top of another, that top cell could actually shadow the bottom cell and prevent light getting through, making this less efficient and lowering the overall hydrogen yield. There's also oxygen produced. So anybody who works with hydrogen knows that you can't mix the two because they will actually combust or explode. So there's a safety risk. And it also has quite a low hydrogen yield. So what about dark fermentation? So in dark fermentation, which is called that way because you are producing the hydrogen under oxygen-free environments, so anaerobic, bacteria such as E. coli and bacillus will actually convert glucose into hydrogen and carbon dioxide. This has a much higher efficiency than the photosynthetic method. And you've also could use a wide variety of carbon sources. So here I'm showing glucose as the carbon source, but in theory, you could use waste water, waste food to make it a more sustainable process. However, there are a few downsides, such as CO2 and other products being produced that you need to separate off from the hydrogen. And there's also quite a low hydrogen yield compared to the more traditional hydrogen production methods that I showed previously. So how can we improve this and increase the hydrogen yield? Well, we can look at genetic modification. So this is the mixed acid fermentation pathway of E. coli, which is one of the metabolic pathways that E. coli use in oxygen-free environments. So we can see hydrogen circled in red at the bottom here. So in order to force E. coli to make more, we need to basically remove other products. We can do this by knocking out particular enzymes or genes involved in the process. So for example, if we remove this enzyme here, PEPC, which converts PEP into oxaloacetate, we remove this entire pathway, and therefore all of our glucose is being converted to pyruvate. We can also knock out these uptake hydrogenases, hydrogenase 1 and 2. They're called uptake hydrogenases because instead of producing hydrogen, they actually convert it into protons and electrons. So if we remove these, we should get higher hydrogen yield. We also have these gene regulators. So these are not enzymes, these are genes that actually control the production of the enzymes involved in this process. So they control it by inhibiting. So if present and activated, they will actually prevent hydrogenase 1 and 2 and the formate hydrogenase lyase complex, FHL, from being made. If we're preventing FHL from being made, formate will stay formate, it will not get converted into hydrogen. So if we knock out these two genes, we get continuous production of the enzyme and therefore continuous production of the hydrogen. Formate doesn't just get converted into hydrogen. There is a pathway where it only gets converted into CO2. So if we knock out the enzymes involved in that pathway, we should get more hydrogen as well. So this is great, but how can we improve this process even better? This is where biocoatings come in. So what is a biocoating? Well, a biocoating is a waterborne colloidal polymer containing live bacteria. We're going to call it a latex now, but it's synthetic latex. So in this coating, the bacteria are non-dividing but remain metabolically active. This means that instead of using some of that glucose to grow and divide, all of it is being used to make hydrogen. So how do we make this biocoating? Well, we start off with a salt-covered scaffold, such as wood, plastic, or even tin foil. So we've coated it with salt, and then we dip this into a suspension containing bacteria and the latex. Now, over time, the salt will disperse through that suspension, and the latex particles will start to film form. You can basically think of this entire process as like a living paint. So a paint is wet when you put it onto something and then it dries. Exact same process here, but instead of using evaporation, we use salt to deform the latex particles. And this means that the bacteria then become trapped in this coating. So why are we producing this coating? I mentioned it has some benefits, but what actually are those benefits? Well, it allows for greater control over the bioreactor environment. You know exactly what is in it. You can prevent contamination to the environment. We are using genetically modified bacteria here, so we want to prevent any contamination to the outside of a bioreactor. We've shown that there's very little escape from these coatings, if none. So therefore, you're preventing any risk of contamination. Likewise, you're also preventing external contaminants such as methanogens, which will actually use hydrogen to convert to make methane, thus reducing your overall hydrogen yield. We don't want those in a bioreactor. But if, for example, a bioreactor containing one of these biocoatings did get contaminated, you would just need to remove the media. You wouldn't actually need to start from scratch. You've already got your biocoating there. Like the bacteria can't get out, bacteria can't get in. So it's just your media that's going to be contaminated. It's also been shown to extend the production life. I mentioned that if all of the energy is going into producing it rather than growing, we can actually have a higher yield. And also one group has shown over 4,000 hours of continuous hydrogen production from a single biocoating. So I'd just like to thank my industrial sponsor, Johnson Matthey, for EBNet for supporting me to present this talk at ISME. And if you would like to find out more about biohydrogen production and biocoatings, you can scan the QR code here and that will take you to my published review article. Thank you so much for listening.